my pleasure this afternoon to be speaking with Dr. Patricio Araos as the finalist for the Stephanie Watts Career Development Award. Congratulations, Patricio.
Thank you.
And thank you for joining us all the way from Chile.
Yes, it's a long trip, but it's a pleasure to be here.
To start off, would you mind telling us a little bit about your background and then also give us an idea about your project pitch that you'll be doing this evening?
Yeah, of course. I'm starting my undergraduate student in biochemistry. I moved from another city to study what is the role of the some treatment like hydrochloric acid and vascular disease and cardiovascular disease, and that was my beginning like a scientist. After that, I did my PhD student in pharmacology in the University of Chile, studying the role of the antigen-presenting cells in the development of hypertension. And after a long time, finished my PhD. Moved for doing my postdoctoral research in the Universidad Autónoma de Chile, but more focused on renal antigen-presenting cells, particularly the dendritic cells, and how they can apparently modulate directly the blood pressure. And that is the project that we are trying to rebuild our own laboratory and that baseline with the renal antigen-presenting cell and how they can modulate different aspects of the blood pressure control.
So dendritic cells are very complex cell types. Do you mind elaborating and expanding on which mechanism within dendritic cells that you're particularly focused on?
Yeah, of course, it's a complicated field, especially when you're talking about renal dendritic cells or renal antigen-presenting cells, because in the kidney, macrophages and dendritic cells share several markers. So it's difficult to say, okay, this cell is dendritic cells, this other is macrophages. Actually, it's very complicated. For us, it's a new field. We're trying to isolate these cells, trying to characterize it for being pretty sure what type of cell they are. The mechanisms that previous data from our laboratory and the collaborators say us that apparently they can modulate the renal sodium handling. And what is the mechanism specifically, we don't know actually. We suppose that the antigen-presenting cells, macrophages or dendritic cells, can secrete some cytokine, of course. However, we don't know actually what is the exactly mechanism. Of course, we all know that there's a different current, especially focused on iso-algae, like Dr. Akirao show us in different papers. But we're trying to move not for that type of mechanism that is more properly in salt-sensitive hypertension. We try to figure out what's happened in salt-resistant hypertension.
So how do you think that this equipment from DSI, if you were to win this award, how could that help your research move forward in the future?
Our results tell us that apparently one of these cells, renal macrophages or renal dendritic cells, the AT1 receptor can modulate this, can modulate the blood pressure in sodium handling or in the vasculature. So we're trying to move for that side to isolate that type of cell, that mechanism, and trying to figure out what's happening in the vasculature in the future. And of course, if we know a little more details about the pathway or the mechanism, could be an additional mechanism that could be answer why the patients in the future can develop hypertension, independent if it's salt-sensitive or salt-resistant.
Very fascinating work. Well, congratulations on being selected as a finalist, and I wish you best of luck for your project pitch this afternoon.
Okay, thank you very much, Macam. Thank you.